And of course, the sunlight will of course provide some support to it. But washing of the garments is the best option rather than keeping it for 3 to 4 days and then reusing it. So I would suggest that we should wash the garments at least after every use. Over to you,
Okay,
Vishal.
thank you. So I think the next question is also related with it, which is from Sushant Sharma, and he says, when a student comes back from school, which means I think he's talking about people who have gone to school, his children, and so on and so forth. How the garments need to be taken care of, and I think you have already answered this question. And basically, I think Ravi and you have taken it up. Make sure your shoes are outside the house. You have a virtual changeover bench, which means the shoes goes off there. Your socks and have to be removed in such a way that you are not touching the outside of the socks is not getting exposed. The way we have shown you about the gloves, how it has to be encapsulated, and the insides are only getting exposed. The cloths and everything have to be removed immediately. Put it in soak them into a detergent solution for at least 6 to 8 hours, and then wash it across. And the child or anybody who's coming from outside should not touch the inside surfaces of the house until unless you have washed your hands thoroughly, which we have shown you the video where you have to spend around 20 minutes scrubbing, cleaning your hands. So I'll move forward from this question. If you have anything to add, Ravi or Ranjit, otherwise I'll move forward.
No, I'd just like to say that, yes, I understand the challenge here. Children are not exactly, I mean, they are exuberant when they come home. I mean, this is a very challenging thing. I mean, this is what we need to wrap our heads around in this new normal that we have. Over to you, Vishal.
Right. And the next question is from Rahul Patil, and he said, can we add bathing when we are in and out of the plant or in and out of the factory? So which means when I'm coming, so I believe I will be carrying some contagion with me, which I would have acquired during my trip from home to factory. So I enter and take bath in the factory, and when I'm reaching back home, I again take a bath. So can we add this kind of bathing as a practice? This is the question for you, Ravi.
Yeah, I mean, see, it depends on the traffic. If you are a very small facility with very staggered timings, maybe a bath can help. But the number of steps which you're again stepping into an alien environment. So I don't see particular value in that. So trying to say that the inside of the factory was dirty, so I have a bath when I come out, or vice versa, it's not really the end of the thing, right? Because both the factory and outside the factory could be dangerous. The only real safe point which we consider for convention's sake is our house. So the point is that as soon as you come from outside, you have a bath and you come into the safe part of your house. But in the factory, you could have a bath and you could end up going into a place where someone has already contaminated, or when you're coming out of the factory, you have a bath and maybe the bus or the car or whatever you have used is reintroducing contamination. So see, bathing cannot hurt, but how practical it is, how much space you have with you, how the persons in the factory will maintain those bathrooms in a manner in which they are, and I just wanted to share one point. This is not a fair thing to ask anyone to do, but if the house has got a spare bathroom, it might make sense to dedicate one bathroom for the people who are going in and out of the house and use the other bathrooms as a regular routine and have a wash down of that bathroom. So the bathroom itself will become a place which is slightly more vulnerable than anything. So you need to be very careful about the bathroom too. I hope I have answered your question. Over to you, Vishal.
Right. Thank you, Ravi. And this is the question which has come from Madhusudan, and he says, "What type of mask we should use?" And Madhu, I'll quickly answer it before passing it across. We have spoken about the mask. What you are supposed to do is it could be a cloth mask, which is good enough because the role of mask is to avoid the droplets which are coming out from somebody who has potential infection to be reaching you. And for that, cloth masks have been proven as effective as any other mask which are being recommended. So any of those things, but very important thing what you have seen in the video is how do you wear a mask and how do you remove a mask. That's more important than the type of the mask, which is important. Next question is from Harshal Uttarwar, and this question is, which surfactant to be used for washing the garments? I'll take this question again. I'll not pass it on because very important thing about surfactants, disinfectants. There is a complete guidance document available which is called as IEST, and they have recommended procedures from 001 to 007, which are dedicated to clean room garments, right from qualification to gowning to holding and cleaning and so on and so forth. So they have some recommended detergents and surfactant which you can use. IEST stands for Institute of Environmental Sciences and Technology. So you can do that, Harshal. I'll move forward. This is again from Sunil Dodamani. It says, is there a necessary steps that need to be taken to push or open the door while entering into primary change room? Again, Sunil, I'll not pass it on. We have kind of complete photographs which we have shown you, and which we says is don't touch anything. I mean, don't touch anything with your hands. Consider everything is contaminated with COVID-19. I mean, that's the mentality you need to go with. And by the way, this mentality is not new to the world. I can tell you, any healthcare professional, when you go to a lab for blood testing, I mean, this blood testing could be a regular testing which you go for, which your company asks you, annual health checkup. And there is a person who takes your blood for either blood sugar and so on and so forth, hemoglobin and other things. Do you know they are taught that everybody who is coming inside is carrying an infection? So they are told that everybody is potential infection, and therefore they don't touch any one of you and any of the things which a patient may be carrying. It could be a perfectly healthy individual. So same thing is there for you. The necessary step of push and open is basically don't touch it. You have an example which was shown. There was a key. There are people who have made it as a foot related activity to open the doors and so on and so forth. So use that, Sunil. I'll move forward, and this is from Dipesh Thawani. And this one is going to you, Ranjit. Could you please explain cleaning of the floor or other surfaces? Is the sanitization required to be IPA based or alcohol based, or any phenyl based solution also helps in cleaning of COVID-19 or removing COVID-19 viruses? Ranjit, this is to you.
Yeah, the flows, of course, as I said in the previous slides, that as of now, the best option, what we have been listening and what we have been going through in the various literatures are either the hydrogen peroxide as well as the sodium hypochlorite, 1% sodium hypochlorite solution. Now, this we in normal case, what we already have in place is either we do clean the surfaces or the floors with a normal disinfectant. But with this pandemic situation and with additional addition of it has become mandatory that we need to ensure or we need to add more cleaning activities in the particular area. So I would suggest that if you are having a disinfectant which has been normally used, please add either hydrogen peroxide or a sodium hypochlorite 1% solution into it, and of course that would really help us in the cleaning of the floors. Over to you, Vishal.
Thank you. And this one again is from Harshal, and he says, what could be the impact of air circulation in the change rooms or on the persons as well as gown? Which means, do we need to do something about air change rate in the area, considering that everybody is a potential for COVID-19? This one is to you, Ravi.
I don't think you need. I don't think you need to tamper with the air conditioning systems, but for sure, office areas are a very, very dangerous place, because a standard package unit in an office is a recirculating unit. There's no fresh air. So please try and work out ways and means to introduce fresh air into your enclosed spaces. As far as areas which are on air handling units, I don't think 20 air changes, which is the base level for a grade D or a support corridor, and then going up all the way to 90 and 100 air changes in grade B areas, I think these are more than adequate to manage the virus. But yes, you need to worry about corridors which are running on package units, worry about offices which are not having ventilation. These are the new things which is going to now we need to consider in a design to ensure that you can introduce air into closed spaces. Over to
Uh,
you, Vishal.
Thank you, Ravi. Just to add on this part, there are studies and which we know from even from a clean room microbiology point of view that if there is the body temperature, what we have is around 37 degrees centigrade, whereas the clean room temperature is around 20 25 degrees centigrade. So what happens is the clean room air is heavy, which is cold, and body air is hot. So the hot air will always go up and it will disperse more, as well as the clean room. I mean, the cold air which is coming from your HVAC or AHU will be heavy and it will go down towards your return razor. And therefore, the entire clean room behavior is being designed based on this hot and cold air, which is coming from body and from the area. Now, very important thing what we also know about it is that if there is no proper ventilation in the room, there is a possibility of these small minute particles to be suspended in the air for infinity. There are studies which were done by White and Niven in somewhere in 1992, based on which where this NBPC counts and all those things started coming into pharma industry, which says that a particle of point, zero point 0.1 micron to 10 micron, they have kept different particles and they have spoken about it. They can take up to 60 days to reach from your plenum of your HEPA to your working height, what we talk about. So therefore, very careful part of it is a proper recirculation and proper ventilation is very important for us. Clean rooms, we do that air change rate calculations and we do those kind of replacement of the air. But as Ravi said, packaging areas, the office spaces where we may not be adding into enough fresh air is something we need to rethink about those kind of things. How do we increase the ventilation over there? Moving forward, this is the question is to Ranjit. When we speak about the shoes, plant shoes, and the factory shoes, this is a question about grade D shoes, which is from Joydeep Das. And he says, instead of using the shoes, can't we use the slippers because that will avoid the sweat? Because if you're wearing a rubber shoes in grade D, there's a lot of sweat which comes up, which increases the contamination. Instead, can we use a slipper? Ranjit?
I would still, yeah, I totally agree. With this, the humid conditions and across India now going forward, there will be always a chance of, the sweating that is coming up. But at the end of the day, what our intention is. So we should be, we should, yeah, there are sites where people are using slippers because that will, and that too, the not exactly slippers, but which is available in the market. That is a Crocs type of shoes, which has openings in the front, and so it can be. But slippers better to Yeah, I totally agree with that, and maybe we can, if we need, we can use the slippers.
Right. So the next one is to you, Ravi, which is from Muhammad Azad Khan. And he says how to handle what type of assessment is to be done due to the absenteeism in the pharma industry. So well, it's being identified that absenteeism is creating a lot of problems. So how do we handle it? What kind of assessments we need to do? Ravi?
Yes, you have spoken about the biggest problem I have on my table right now. That trying to get people into the factory is been a huge challenge. Even though you are working in an industry which permits people to transit, we are considered an essential industry. But there are these, let's say, households, housing societies, even villages which are neighboring from where our manpower comes, where there is a gram panchayat and the pradhan. All these people have now, I mean, society chairman, all these people have become, let's say, they have got a certain level of power and they have started enforcing strange rules which sometimes block people from coming to work. We've had these complaints from our staff, and frankly, what we've been doing is we've been operating at 70%. That's the only way, because you cannot understaff the situation and have a problem, in manufacturing. So I can speak of our approach, which has been to understaff. I'm sorry, use a lower rate of production to manage it. And that's the way we are working. So I have nothing else to offer for you. Yes, over to you.
Okay,
Shall I
so
add some points
yeah,
here?
please go ahead. Go ahead, Ranjit.
So Vishal, what we are doing is, because we are also, as you know, everyone is aware of what the situation is in Gujarat and Ahmedabad, the leading from the front. As of now, as at least Mr. Ravi is at least lucky that they are running with 70%. My attendance yesterday was 43%, and with 43%, we need to run the show, because being a manufacturer of oncology, we know that the oncology drugs are very much important, and for those patients which are really, they're almost they really need that. So what we have done is that we did a risk assessment for majority of the things. Maybe found that, let's say, for example, take the example of calibration, which has to be done by an external agency. Do we really need to do that? Because everywhere it is locked down. So we took a temporary change control sort of a document with all this information, whether this calibration is required or preventive maintenance from external resources. And of course, there were many days where we were running short of manpower in the quality control lab, and of course in production. So in QC, again, the temporary change control played a very important role with all the risk assessment for whether we really need to do the complete analysis in API as per the specification, or we can at least reduce the going forward with the reduced testing for excipients. So these type of things we try to inculcate with a proper document, risk assessment, getting the approval from the quality and regulatory, taking the approval from the regions wherever the product is going. For example, if the product is going to Europe, we have taken the approval from the QP. So these many things has helped us to to avoid a little bit. Of course, there is the situation is not so good, but at least rather than switching off the line or keeping the entire site idle, we are at least running the show with a few batches at least on a weekly basis. So the temporary change control, and everyone is aware, no one is going to, if you have not done the analysis in this situation, no one is going to do, come and give you a 483 or asking you to initiate an FAR. No, but of course, there has to be a proper document with a proper risk assessment, and of course, that's that will help us in not only anyone will be helpful with that particular document, but the risk assessment and the document should be available, providing that there is no risk.
Right. Thank you so much, Ranjit. That's very detailed. This question is again for you, Ranjit, and this is about cleaning and sanitization. And this is being asked by Dipesh Havani, and he says, how many times sanitization is recommended in the office and other parts of the plant, which is, I believe, is CNC areas, like change rooms he's asked about, where the people movement is more. So plant entry, change rooms, and so on and so forth. How many times sanitization should be done? Is it every shift? Is it twice a shift? Or what is it?
See, Vishal, in my previous one of the slides, it was already shared, but it was for other areas. Again, the change rooms, wherever the primary, the factory gowns are worn, or where the traffic is more. Again, it's there is no defined set of frequency. One has to identify the amount of the traffic that is in that particular area. But if you are having a frequency of once in a shift, I would suggest go for at least 2 to 3, at least two more cleaning in between after maybe every 2 hours or 3 hours, so that overall bioburden in that particular area and the flows and the lockers, everything is under control. So it's again, it depends upon the person, it depends upon the location, it depends upon the traffic. So many things comes into picture, and not can't say that it has to be if you're doing it for once in a shift, you increase to 3 times or 4 times. So many things and many factors play a role in that.
Okay. So this one is from Palash Das, and this is to you, Ravi. Before changing any SOPs drastically, if we want to perform a protocol-based assessment of the entire facility, then what are the points which we need to consider during this assessment?
That's a pretty sweeping question. I mean, it depends on which procedure you want to change, how critical it is. So I don't know exactly how you want me to field that answer. Do you want to jump in, guys, any of you? Because I cannot give you a general answer to that, yes, these are the points to check. I can take an example. Let's take calibration. Ranjit just mentioned that. Now, one method would be that you look at old calibration data and find out how many times there was a need to put in an offset within the, if you have a calibration for a temperature, let's say you calibrate at 30 degrees and 150 degrees and expect the autoclave to function at 121. So you verify at 121. So once you've done the slope check at 30 and 150 and you want to verify at 121 and your protocol says plus or minus zero point 0.5 degrees is okay, you don't put an offset. If the difference you have found at the end of calibration is a little more than 0.5 degree, you put in an offset. Now you can look at what you had in the last 2 years or 3 years, whether you had to put in offsets or not. And then you can take a call that, okay, even when I put in my offset, the difference was only 0.1 degrees. So, okay, as long as my temperature, which is set to run at plus or minus 1 degree, is buffered by the experience of the last 3 years, I can extend my calibration frequency by 3 months, maybe, just so that I accommodate this COVID situation where my calibration vendor is locked out in some other city or something like this. So this is an example of how you would consider a risk-based approach to understand how you can change a procedure. And there is no protocol per se for this because you are looking at old data. So if you want to write a protocol which says, look at old data, record it in this manner, and then write, okay, so be it. I mean, that can also be done. I'm not sure I've answered your question correctly, but yeah, over to you, Vishal. I think we have hardly a few minutes left to go, so we need to be quick. Yeah,
Sure.
over to you.
Yeah. So thank you, and I'll ask. We know Harun Siddiqui, who is also in this webinar, and Harun heads the engineering and projects department of Mylan Injectables globally. So Harun, what's your point, or what's your take on this when we talk about the changes which we need to look at, or changing the things with respect to engineering, with respect to calibrations, with respect to maintenance, and so on and so forth, bringing people from outside? What do you foresee are the changes one, or what are the points one need to consider before making those changes? And Bini, can you unmute Harun? Harun, are you there?
I think Vishal, you've run out of time, so I'm not sure what is happening because I can.
Hello,
Okay, so I think yes,
can you
Harun.
get me?
Welcome.
Yeah.
Yes, we can.
Basically,
Welcome.
no. Basically, I'm a director with like 2 companies, and I don't look after the engineering part, but I look after the entire part, including the audits. So I was very keen to go through with you people, but I suppose in this clean rooms sessions that you all have come up with, they've been very nice. But on the practical part, to get a lay laborer who goes in on a shift basis is going to be a challenge for us, and especially operating at 30%, 70%, 40% at different times. So there should be something which is more robust, change controls which have to be put in, need to be put in, or deviations to be recorded. I still feel that the temperatures gun that we use is useless. We cannot douse everyone in hypochlorite. So going into that, we have to change all these type of taps that we have and make them all, automatic, so that we don't have to touch anything. We have to have stands basically which we can use as from a footrest. So there are a lot of things which, okay, fine, you're doing it in a very short time, which I feel may not have been covered, but we can definitely go for these type of improvements. Like we used to have hand dryers. Now today I would not recommend a hand dryer in this condition.
Wonderful. I think, thank you, Harun. I think it's very profound. And the idea is that we start this discussion and we bring in a lot of these kind of good practices where we all collaborate rather than just a presentation. So we will collect a lot of information, keep on sharing with everybody. There are several organizations and scientific bodies who are working on it, but we should work on that particular part. Good. I'll take the last question, and this is the question which is very pertinent question, and it is about environmental monitoring. And this says, is there any idea of monitoring environment for coronaviruses by swabs based on facility and biological products susceptibility to virus? So this is the last question, which we'll take it. So we know environmental monitoring, we do it by swab sampling, we do it by air sampling, and we do it by shuttle plate. So the question is, can we do something by for environmental monitoring for viruses also? I'll give it to you, Ravi, this question.
Okay, so we have struggled for several years to, monitor the presence of, we make measles virus. And we have tried to detect measles virus in our areas in order to manage campaign manufacturing. And we have not succeeded because the methods available with us do not detect down to such small levels where, there can be, one or two virus particles. So I think this is early days. We don't have any methods in which you can really capture or detect from the air, because that requires a sort of panic level. I'm sorry, that's got some echo. Yeah. So I don't think we have any methods currently to do this.